Okay, so welcome back friends. Welcome back in another video tutorial from microbiology with Chaglik. In this video tutorial, we will going to discuss about some antibiotic who specifically inhibits the cell wall synthesis. Okay, so as like our previous video, we will going to first discuss about some basic description of cell walls. Then we will going to discuss about how we can inhibit them. So first I would if you want to take a screenshot of that difference, that is the gram positive cell wall difference and gram negative cell wall difference, then you can take. We will going to discuss about this diagrammatically. So this is a diagram of gram positive bacterial cell wall and this is a gram negative bacterial cell wall. So gram positive bacterial cell wall has a thick peptidoglycan layer. Okay. On the other hand, the peptidoglycan layer in case of gram negative bacteria is thin. Okay, but in turn, gram negative bacteria also possess a outer membrane, which gram positive bacteria lacks. Okay. Now, again, gram negative bacteria have some proteins and lipopolysaccharides, which gram positive bacteria lacks. Okay, and I want to told you about that gram positive bacteria can develop less antibiotic resistance as compared to gram negative bacteria. That means most of the gram negative bacteria are antibiotic resistance. Gram positive bacteria are very rarely show antibiotic resistance. Okay, so now after discuss about this, I want to discuss with you about the various components of the peptidoglycan, okay, along with their structure. So peptidoglycan consists of two carbohydrate structure. One is NAM, that is N-acetyl muramic acid, and NAG, that is N-acetyl glucosamine. Okay, so N-acetyl muramic acid, N-acetyl glucosamine, who are joined by a glycosidic bond. Because they are carbohydrates, so it is obvious that they will be joined by glycosidic bond, and in between them there is a two kind of pentapeptides present that are joined together by peptide bond. Okay, so that is how the construction of peptidoglycan looks like. So they are consist of NAM and NAG, who are joined together by glycosidic bond, and inside them they have pentapeptides, right, who are joined together by peptide bonds. Okay, so now we will going to discuss about various kind of antibiotics. Okay, so first antibiotic that I want to discuss with you falls on the class of beta lactamase, that is beta lactams, not lactamase, beta lactams, that is penicillin. So first it is important to discuss about the structure of the beta lactams. So you will get a NH here and here a carbonyl. So that is the structure of beta lactamase. Okay, a 5 carbon ring and a 4 carbon ring, and inside that you have a NH here and a C double bond O. Okay, so the first one that we want to discuss is penicillin. Okay, so let us discuss. So first of all, I want to discuss with you about a binding protein named penicillin binding protein, which is already present on the cell membrane of the bacteria. That is, it is a membrane associated protein. Okay, and this protein name is penicillin binding protein. So now whenever we will add penicillin, this penicillin will bind over here. Okay, so upon binding with the penicillin binding protein, it stimulates penicillin binding protein. Okay, and as a result, this stimulated penicillin binding protein will activate these scissors. Okay, so these scissors are nothing but autolytic enzyme. Okay, so this autolytic enzyme will causes the lysis of or the destruction of peptide bonds and those glycopeptide bonds. Okay, so again one more time. So what is going on here? Whenever penicillin will bind over penicillin binding protein, the penicillin binding protein becomes stimulated, okay, and as a result they in turn activates to these seizures, which is known as autolytic enzyme. So this autolytic enzyme will destroy all the peptide bonds and glycosidic bonds for you, and as a result cell wall becomes disrupted, okay. So that is the number one mechanism by which penicillin works. And the second mechanism is, upon binding with penicillin binding protein, penicillin activates transpeptide enzyme. So they will activate the transpeptidase enzyme. So now what is the function of transpeptidase enzyme? So let me discuss with you that transpeptidase enzyme have the ability to cross-link those pentapeptides. That is why they are how goodly arranged, right? So if penicillin binding protein inhibits that transpeptidase enzyme, then what will happen? So if penicillin binding protein inhibits transpeptide enzyme, as a result the cross-linking will be inhibited, and which ultimately destroys the cell wall, okay. So that is how the two mechanism penicillin follows for the disruption of the cell wall. Number one is already obviously the autolytic enzyme. This autolytic enzyme will cleave the peptide bonds as well as glycosidic bonds, and the number two mechanism is transpeptidase enzyme inhibition. This transpeptidase enzyme involved in the cross-linking of those pentapeptides, and as a result the cell wall will be disrupted, okay. So now what kind of bacteria can be treated with the help of this penicillin? So any kind of gram positive and gram negative bacteria was previously possible to treat with the help of penicillin. But now let me introduce a new problem here. So what is those problems? So there can be a possibility that bacteria can contain plasmid, right? We know about this, right? So now under such circumstances, if that plasmid contain gene, which gene encodes a special kind of enzyme known as beta lactamase, under such circumstances that particular bacteria can develop resistance to penicillin. Let's discuss how. Because the beta lactamase, as its name suggests, it has the ability to destroy the beta lactam ring, okay. So as a result what will happen? I have told you that they are all beta lactams antibiotic, right? So if beta lactamase enzyme can be secreted, or if bacteria have the ability to secrete beta lactamase enzyme, so bacteria can protect themselves from that penicillin by simply destroying that beta lactams. And if it become destroyed, can it bind with that penicillin binding protein? No, obviously not. And as a result penicillin cannot help us to treat those kind of infection, okay. So this kind of situation is mostly observed in case of gram negative bacteria. That is why penicillin are nowadays used only for gram positive bacteria, okay. For example, streptococcus infection or any kind of syphilis infection, okay, are treated by penicillin. So that was all about penicillin. Now let's discuss about another important drug, that is amoxicillin, okay. So I want to told you that as it is in the same class with penicillin, it follows the same mechanism of action what penicillin follows. That means it will again activate autolytic enzyme upon binding with penicillin binding protein, and it will also inhibit transpeptidase enzyme and prevents the cross linking, right? So now I want to discuss with you another important property of amoxicillin, that is amoxicillin will not show its action if it is used individually, okay. That means I want to discuss with you that amoxicillin or the beta lactam ring which is present in amoxicillin is highly susceptible to beta lactamase, okay. That is why whenever you are using amoxicillin, you have to use another kind of chemical compound named clavulanic acid. This clavulanic acid have the ability to act as a beta lactam beta lactamase inhibitor. Okay, so whenever you want to use amoxicillin for any kind of treatment, you have to use along with this clavulanic acid. Okay, so any kind of respiratory tract infection, let's say any kind of meningitis or urinary tract infection, we are using amoxicillin. Okay, and also for middle ear infection, otitis media, we are also using amoxicillin, but addition of clavulanic acid along with amoxicillin is necessary. Okay, now let's discuss about another kind of antibiotic which is also under beta lactams, that is cephalosporin. Okay, so there are total four important generation of the cephalosporin. The one is generation 1, generation 2, generation 3, generation 4. So let me discuss with you that the first two generation, that is generation 1 and generation 2, is specific for gram positive bacteria infection. Okay, let's say any kind of streptococcus infection, or for example streptococcus pyogenes or staphylococcal aureus. Okay, and the next two, that is generation 3 and generation 4, is strictly for gram negative bacteria. For example, gram negative Pseudomonas aeruginosa, Klebsiella pneumonia infection, bacteria causing meningitis infection, and different type of Enterobacter. Okay, so what is the first two generation, that is generation 1 and generation 2, we are using for gram positive bacteria. For example, streptococcus pyogenes and staphylococcal aureus. And generation 3 and generation 4 is strictly for gram negative bacteria. For example, gram negative Enterobacter, Pseudomonas aeruginosa, bacteria causing meningitis. Okay, so for example of generation 1 antibiotic is cefalaxin. Cefonicid is the example of generation 2, cefotaxime is the example of generation 3, and cefepime is the example of generation 4, although other examples are also present. So main important thing to remember here, that the generation 1 and generation 2 of cephalosporin is strictly for gram positive bacteria. For example, streptococcus aureus, staphylococcal pyogenes infection. And generation 3 and generation 4 is strictly for gram negative bacteria infection. For example, gram negative Pseudomonas aeruginosa, Klebsiella, bacteria causing meningitis infection, and different type of Enterobacter infection. Okay, so this is the most three important mode of action of beta lactam antibiotic: penicillin, amoxicillin, and cephalosporin. Okay, along with another important antibiotics are also present, for example carbapenem and penicillin derivative. Now let us discuss about another important but other kind of antibiotics that is fall on the glycopeptide group. Okay, so most important example here is the vancomycin. So another important thing, that the previous three type of antibiotics that we have discussed, penicillin, amoxicillin, cephalosporin, are bactericidal. Bactericidal means they will ensure the death of the bacteria. Yes, obviously, because if cell wall gets disrupted, there will be too much amount of water and ions which will leaking in, right? And as a result, cell lysis will occur. Okay, now let's discuss about the function of the vancomycin. So vancomycin will bind with the carboxyl terminal of a pentapeptide, and they will inhibit the peptide bond formation between two pentapeptides. Okay, and as a result, what will happen? The cross-linking between pentapeptides will be hampered, and which will in turn facilitate cell wall disruption and cell lysis. Okay, so vancomycin is useful for methicillin-resistant staphylococcal aureus treatment, gram-positive bacterial infection treatment, and Clostridium difficile infection treatment. Okay, and also some resistant coagulase negative bacteria treatment. Okay, so MRSA, methicillin resistant staphylococcal aureus, we will going to discuss about this in our upcoming videos, along with gram positive bacteria infection. The number 3 is resistant coagulase negative bacteria infection and Clostridium difficile. Okay, so dalbavancin, oritavancin along with telavancin are also important example of glycopeptides. Okay, so another important thing that is important to discuss is the isoniazid type of antibiotic. Okay, although it is specifically applicable for only a particular kind of the cell wall that you may find in Mycobacterium tuberculosis. Because remember it very carefully, I will also discuss with you about this in details when we will going to discuss about tuberculosis disease. That is, Mycobacterium tuberculosis cell wall is rich in a particular type of lipid. Okay, that is quite unusual because we are usually facing any kind of polypeptide or lipopolysaccharide as a component of the cell wall, but in that case you will get high amount of lipid in cell wall. So the example of that the name of that lipid is mycolic acid. Okay, so cell wall of Mycobacterium tuberculosis is consist of mycolic acid. Okay, and it is produced by a enzyme which is activated by a long signaling pathway that we will not going to discuss. It is extremely difficult, and as a result the content become too lengthy. We will not going to discuss with about this, but we will going to discuss about that isoniazid will block the mycolic acid synthase enzyme. Okay, and as a result it prevent the mycolic acid synthesis, which ultimately causes disruption of the cell wall. So in the first antibiotic that we have discussed is beta-lactam, a type of antibiotic. So among beta-lactam, the first that we have discussed is the penicillin. So penicillin can activate autolytic enzyme and inhibit transpeptidase enzyme upon binding with the penicillin binding protein, and that's how they causes cell wall disruption. We have to keep a very important thing in our mind, that is beta-lactam antibiotic resistance, right? That is acquired by microorganism when they have a plasmid which contains gene that can produce beta-lactamase enzyme, who have the capability to destroy the beta-lactam antibiotic. Okay, so that is why penicillin is mostly useful for gram-positive bacterial treatment. We have discussed about amoxicillin. If we use amoxicillin along with clavulanic acid, only when amoxicillin can show its activity, so it can help us to treat any kind of respiratory tract infection, meningitis treatment, any kind of bacterial urinary tract infection, and middle ear infection called otitis media. We have discussed about cephalosporin. So there are main important 4th generation that we have to discuss, and among them the first 2 generation is strictly for gram-positive bacteria, for example Streptococcus pyogenes, Staphylococcus aureus, and the second 2 are strictly for gram-negative bacteria, for example Pseudomonas aeruginosa, Klebsiella pneumonia infection, bacteria causing meningitis. And we have also discussed about vancomycin. This vancomycin is strictly inhibiting strictly blocking the carboxyl end of a pentapeptide, and as a result it prevents the formation of peptide bond with the another N-terminal end of the pentapeptide, and which prevents the cross-linking of the pentapeptide, right? And as a result cell wall gets disrupted, and which facilitates cell lysis. It is mainly used for methicillin resistant staphylococcal infection treatment, along with some gram-positive bacterial infection treatment, Clostridium difficile treatment, and resistant coagulase negative bacteria treatment. Okay, and at last we have discussed about isoniazid. This isoniazid simply inhibiting the mycolic acid synthase enzyme. Mycolic acid synthase enzyme, not synthesis, synthase. This mycolic acid synthase enzyme will, as it gets inhibited, it can no longer produce mycolic acid, and which is the important component of the cell wall, and as a result the cell wall of the Mycobacterium tuberculosis become disrupted, right? Also there are various kind of resistant mycolic acid present, resistant mycobacteria present, that are commonly known as MDR-TB, have the ability to show resistance towards isoniazid, and under such circumstances we have to use a mixture of 4 drugs named isoniazid, rifampicin, ethambutol, and pyrazinamide. We will going to discuss about those kind of things detailedly in case of when we will going to discuss about tuberculosis disease. So that's all for today, friends. So today we have completed the cell wall inhibitors. Okay, so we will again come back with another video where we will going to discuss about protein synthesis inhibitor. Okay, and then we will going to discuss about cell membrane inhibitor, and then we will complete our lecture series, right? So thank you for listening to this class.